Hello everyone, I am Dr. Khushboo, Assistant Professor, Microbiology, School of Basic Sciences, and today I am going to present a lecture on Introduction to Microbiology. So here are some contents which we are going to see throughout this PPT. So firstly, did you know that you are all microorganisms more than your cells on your body? Microorganisms are present all over your body, inside your body, and in the river, in ponds, in air, and in the rocks. So they are unipresent. So the microorganisms, the study of microbiology is revolve around the study of microorganisms. Microorganisms are the smallest bacteria, and these are having the smallest size which cannot be seen through the naked eyes. Microbiology revolves around the two themes. First, understanding the basic life processes, and second, applying that knowledge to benefit of the human's life. So understanding the basic life processes means how microorganisms are excellent, how models are there present in the nature, and understanding their cellular processes, and how we can apply that knowledge to the benefit of the humans by knowing their roles in the medicines, agriculture, and industries. So in this slide you can see over here this microorganism classification. Earlier, microorganism classification was very difficult for the taxonomist. So they classify the organism into main two categories, two domains. First, the human, and second is plant. But after that, they recognize that the microorganisms are having some capabilities or properties to move, means they are showing animal-like properties, and as well as they are having the plant-like properties, like they are photosynthetic and they can produce more oxygen than the plant in the planet. So further they are categorized into 5 domains. 1. The Monera, 2nd Protista, 3rd Fungi, 4th Animalia, and 5th Plantae. So the Monera is the prokaryote, and the other four were categorized under the eukaryotes. Prokaryote means the prokaryote word is made up of 2 words. So first, pro means the earlier, and karyotes means the nucleus. It means these are the microorganisms which are having primordial nucleus. That means nucleus without nuclear membrane. And the eukaryote means the U means here it is a new, and karyote means nucleus. It means those microorganisms having the new nucleus, means the nucleus having the nuclear membrane around it. Under this, the viruses are not classified. Why? Because viruses are acellular entities, and these are not classified under these domains or kingdoms. They are 10,000 times smaller than the bacteria, and this smaller size of the viruses is the very helpful to those to cause many kind of diseases in the human and plants. They are the game changer in the whole world. Nowadays you can see that viruses are the main problem creating in the disease in the humans. So they invade the host cell in order to replicate in them by using their machinery and causing the disease, which can be communicable as well. Now in this slide you can see there are many fields of the microbiology. Some are basic, some are applied. So in the basic fields of microbiology you can see virology. Virology is the study of viruses. Bacteriology, the study of the bacteria is done. Then phycology, the study of algae. Mycology, study of fungi. Protozoology, study of the protozoas. And some are the applied sciences or applied field of microbiology, like medical microbiology, In this, including the immunology, which deal with the immunity system of the human, which provide us or protect us from the various kind of diseases present in the nature. Then food and dairy microbiology, that deals with a different type of manufacturing of food products or dairy products in the industries. Then coming to the agricultural microbiology, that including the crop yield and diseases dealt with the plants, which are the microorganism in this, helping how to combat with these diseases, those causing the low yield of the crop. Then industrial microbiology. In this field, microbiologists deal with the production of the various kind of the vaccines, antibiotics and steroids, etc. Then public health microbiology, that deal with the study of the epidemiology of different kind of diseases in the human. Now, if we are seeing we are studying about the microbiology, so how can we see the microorganism? It is the question which is mainly arises. So to see the microorganism, those are very small entity which cannot be seen with the naked eyes. We need some kind of instrument. So that instrument is known as microscope. Here you can see the picture of the microscope present on your side. So anything less than 1 millimeter must be examined through the microscope, and several scientists contributed the discovery of microscopes, just like you can see over here, Jonathan Janssen in 1590, Galileo Galilei in 1609 and Robert Hooke in 1660. They all contributed in one way or another to development of the microscope. Here in this picture, you can see that the microscope which is present is a compound microscope. So there are different type of microscope present. They can be simple microscope, they can be compound microscope, and nowadays we are having are with us scanning electron microscope, which is very specific and having very high resolution than the simple and compound microscope, which can be very useful in the field. Let's come to some historical perspectives. Now, firstly, we are having here Anton van Leeuwenhoek. In 1632 to 1723, he is the father of microbiology and also considered as the first microbiologist. Leeuwenhoek made more than 500 optical lenses in his lifetime, and he also created at least 25 microscopes, and out of these all are different, and out of which only 9 were survived, hopefully. So his single lens magnified 50 to 300 times of magnification, and between 1674 to 1723 he wrote series of paper describing his observations of bacteria, algae and protozoa and fungi. Those are called by himself animalcules. Here in this picture, you can see over the drawings of the Anton van Leeuwenhoek provided of his microscopes, and the second one is the simplest microscope made by himself, which is having a simple convex lenses and the handle to focus the microscope. There is a focus knob over here. Then after the Anton van Leeuwenhoek, here comes the Robert Hooke. In 1635 to 1703, he reconfirmed Anton van Leeuwenhoek's discovery of existence of the tiny living organism. One of the most important discovery of biology occurred in 1665. In that era, he helped the crude microscopes. With the help of the crude microscope, Robert Hooke stated that lives of the smallest structural unit were the cells. He was the one who developed the cell theory. He presented or published his drawings of the different kind of microorganisms and cells in the his well-known book Micrographia. In this picture, you can see there is a cell structure of the cork which was seen by the Robert Hooke in 1665 and known as these smallest creatures, these smallest cells are known as tiny rooms by him. The next is coming to the theory of spontaneous generation. Earlier, Aristotle, a great philosopher, he postulated this theory, the theory of spontaneous generation. According to him, he said that, or he having the belief that life could arise from the vital forces presented in the non-livings or decomposing matter, that is known as abiogenesis. According to him, the life can originate from the non-living things if that matter having the vital heat in it. So in the other words, organisms can arise from the non-living matter as he is considered. But after that, Francisco Reyes came into knowledge and he set the experiment and consisting of the open containers. And he uses the three containers. One was the open, the second was the sealed with the cork, and third container was covered with the mesh. And he allowed air to let in those containers. But accordingly, the open container having full source of air, that air contaminated the meat and having the flies on it, and flies came and maggots were generated over him. In the second, the corked tube or corked container, the flies cannot let into the meat. So there were no origination of the maggot in that. And the third was the container which was having the mesh over it. So flies were having their maggots or eggs on the top of the mesh. So it has already suggested by the Francisco Reyes and his experiment that if the flies cannot contact into the meat, it cannot be originate life from any other thing. So it was further disproved by Schwann, Frederick Schrödinger and von Deutsch 1830s. Air allowed to enter the flask, but only after passing through the heated tube or sterile wool. According to John Tyndall in 1820 and 1893, he did the further experiment. He omissioned the dust to enter into the test tubes so that there will no growth. So he demonstrated that if there is no dust or air coming into the test tube, there will be no sign of origination of any bacteria. So after that, he also demonstrated some kind of heat resistant forms of bacteria. Those are known as endospores. Now further, we are having with us Louis Pasteur, 1822 to 1895. He is a great scientist in the microbiology field, also known as the secondly the father of modern microbiology. He disapproved the spontaneous generation of microbes by preventing the dust particles, as the Tyndall did, from reaching the sterile broth. He used the different type of necks in the beakers. In 1861, completes experiments that lays to rest in spontaneous generation. So he showed microbes caused fermentation and spoilage too. In this picture, you can see in his experiment, the microorganism get trapped into the airborne organism into the cotton, and he also heated the necks of the flasks and drawing them into the or out to the long curves. So sterilized media he used in it and left the flask open into the air. Because the media was sterilized and the neck was heated, which was passing through the neck is going to sterilize without any microorganism. So there was no microorganism passed or put into the flask, so it will not going to cause any kind of spontaneous generation. In this slide, you can see there are other important contribution of Louis Pasteur. As I already told you that in the field of microbiology, Louis Pasteur is also known as the father of modern microbiology, and it is because he did various kind of experiments, and his contributions are well known in the field of microbiology. So he coined the term microbiology. He was the one who noted this study should known as the microbiology. Then he proposed germ theory of diseases. Then he developed sterilization techniques by which he proposed that media or our microorganism can be supposed to prepare or sterilize the further. Then develop methods and techniques for the cultivation of microorganisms. Studies on pebrine, that is silkworm diseases, and anthrax, cholera, hydrophobia is done by himself. Then he proposed the theory of pasteurization, which is nowadays very well known, very well accepted in the industrial microbiology and food and fermentation industries. Then he coined the term vaccine. After this, we know about the Heinrich Hermann Robert Koch. He was a very well-known scientist, 1843 to 1910. He demonstrated the link between the microbes and infectious diseases. He was the one who identified causative agent of anthrax, cholera, and tuberculosis. You have to note that he identified the causative agent of these diseases. And he is regarded as one of the main founders of modern bacteriology. And he is also known as father of bacteriology. So he was the first to use an oil immersion lenses and condenser that enabled smaller object to be seen. So developed techniques the solid media. He also developed the technique of the solid media. He used the gelatin in the media to solidify the medias for obtaining the pure culture of the microorganisms. So some still in existence today. And he was also awarded Nobel Prize for Physiology and Medicines in 1905. So one of the major contribution of the Heinrich Hermann Robert Koch was his Koch postulates. The Koch postulate nowadays are very, very well known, and this is the one postulate which give us the different kind of to study, which are helpful to study the different kind of diseases and to help in the production of their vaccines and antibiotics. Here are some other contributors. Edward Jenner in 1796. He vaccinates people to protect them from smallpox. In 1876, German botanist Ferdinand Cohen also discovered heat-resistant forms of bacteria. Those are known as endospores. This term bacteria is called endospores, and he found that the specific Bacillus species and Clostridium species at his time. Then Joseph Lister, 1827 to 1912, he developed a system of surgery designed to prevent the microorganisms from entering wound, and phenol, that is carbolic acid, sprayed in the air around surgical incision. So this decreased the number of the post-operative infections in the patient, and he published his findings in 1867 and transformed the practice of the surgery at that time. Then Paul Ehrlich in 1890s, Ehrlich proposed a theory of immunity in which the antibodies were responsible for immunity. That means they acting as antitoxins. So in addition, he known as father of modern chemotherapy. He speculated about some magic bullets that would selectively find out and destroy the pathogens which are present in the body or coming out from the outside, but not harm the host specifically, and thus giving the idea of selective toxicity in the body. So he also developed a staining procedure to identify tuberculosis bacilli, which are the causing agent of tuberculosis disease. Here are some more which we have to see, that is Alexander Fleming. Alexander Fleming in 1928 observed that the growth of bacterium, that is Staphylococcus aureus, was inhibited in the areas surrounding the colony of mold, and that had contaminated a petri plate. And it was a mere case of surprise for him. The mold was identified as Penicillium notatum, and its active compound was named penicillin. Then Walter Hess, 1846 to 1911. He used agar first time. He was the first one who used agar as a solidifying agent. Before Walter Hess, the solid medium was maintained by use of gelatin, and agar is the first one is used as a solidifying agent to harden the media, and agar is extracted from the seaweed, that is red algae. Then Richard Petrie, 1852 to 1921, he used agar dish to provide a large area to grow. It means he was the one who discovered the Petri dish, and nowadays Petri dishes are very important tool in the microbiology. So Christian Gram, 1853 to 1935, he was the one who developed the staining techniques in the microbiology, and staining method that demonstrated the bacteria and distinguished bacteria between gram positive and gram negative. Then Raymond Sabrod, 1890 to 1910, developed the culture media to study yeast and mold. Then Dmitry Ivanovsky, 1892, he gave the tobacco mosaic virus could pass through the filters through the removed bacteria. Then Selman Boxman in 1940, he discovered a number of antibiotics such as tetracycline and streptomycins. Here are some applications of microbiology. The microbiology can have many applications, and several points are here, you can see over here. The recombinant DNA technology. Genes in the microbes, plants, animals manipulated for their practical application. For example, production of human blood clotting factors by E. coli to aid hemophiliacs. Then in case of gene therapy, inserting a missing gene or repairing a defective one in the human by inserting desired gene into host cell to produce important therapeutic substance such as insulin, human growth hormone, and etc. Then in case of environment, recycling carbon, nitrogen, phosphorus, and many other gases. Microorganisms play a vital role to maintain the geochemical cycles. Then bioremediation, live bacteria, fungi, algae, these can use to detoxify the polluted environment. Then in case of food fermentation industries, to produce the food from the brewing, wine and other, these can use to produce cheese, etc. So after that, coming to the research, you can see over here that these microorganisms having the most simplest life process and can be used to doing the many researches. So here are some future scope of the microbiology. Future challenges such as finding new ways to combat the diseases, reduce the pollution, and feed the world population is one of the major challenge for the microbiology. Studying the role of microbiology as a source of high quality food, other practical products such as enzyme production in the industrial application is second. Then host pathogen relationships, degrading the pollutants and toxic waste, and use those microorganisms as a vector to treat the diseases and enhancing the agricultural productivity. Here are some references. Thank you so much.